Fertilization is the process by which the two gametes from the parents fuse their genetic material to form a new individual. Fertilization requires that sperm cells, swimming through the uterine tube, contact a secondary oocyte. Once the sperm make contact with the oocyte and penetrate through the cells of the corona radiata, they enter the area of the zona pellucida. A glycoprotein in the zona pellucida triggers release of digestive enzymes from the head of the sperm. These enzymes help to digest a path for the sperm to reach the plasma membrane of the secondary oocyte. Syngamy occurs when the first sperm to reach the plasma membrane fuses with the secondary oocyte. After a sperm penetrates the cell, the secondary oocyte completes meiosis II. The oocyte subsequently divides into a larger ovum and a smaller secondary polar body that fragments and degenerates. The 1st through 8th weeks after fertilization are called the embryonic period. During the first day following fertilization, the zygote begins mitotic cell divisions called cleavage as it moves through the uterine tube. The cells formed are called blastomeres. By the 4th day after fertilization, the blastomeres have formed a solid ball, now called a morula. By day 4 or 5, the morula enters the uterine cavity. Here, glycogen-rich secretions from the endometrial glands nourish the developing morula. The blastomeres in the morula are stimulated to rearrange themselves and form a fluid-filled hollow ball of cells known as the blastocyst. The cells continue to rearrange, forming an area of more cell layers called the inner cell mass. The blastocyst's outer layer of cells is called the trophoblast. About 6 days after fertilization, the blastocyst attaches to the endometrium of the uterine wall. During the 2nd week after fertilization, the implanted blastocysts differentiate into 2 layers. Together, the hypoblast and epiblast layers form the flat bilaminar embryonic disc. During the 9th day, the epiblast cells multiply and migrate, forming the fluid-filled amniotic cavity. At the same time, the multiplying hypoblast cells begin to develop into a yolk sac. As a result, the bilaminar embryonic disc is positioned between the amnion and yolk sac. By the 12th day, differentiation of yolk sac cells forms an extraembryonic mesoderm. The migrating yolk sac cells, as well as the syncytiotrophoblast and cytotrophoblast layers, form the chorion. The chorion grows to surround the amnion and amniotic fluid and forms the connecting stalk, which is the future umbilicus. The chorion, destined to form the embryonic part of the placenta, is important in the exchange of nutrients between maternal blood and the embryo. During the 3rd week after fertilization, the 3 primary germ layers are formed, which provide cells for organ formation in the following months. These germ cell layers are formed by a process known as gastrulation, which involves rearranging epiblast cells. As cells from the epiblast migrate, a faint groove, called the primitive streak, forms on the surface of the embryonic disc. With the formation of the primitive streak, it is possible to recognize the head and tail ends of the embryo. Some epiblast cells migrate, differentiate, and then replace the hypoblast to form the endoderm germ layer. Other epiblast cells migrate and differentiate to form a new layer between the epiblast and hypoblast, identified as the mesoderm germ layer. The remaining epiblast cells become the ectoderm germ layer. The result of gastrulation is the formation of the trilaminar embryonic disc, consisting of the 3 germ layers. The ectoderm gives rise to all nervous tissue and the epithelium of the skin. The mesoderm gives rise to blood, muscle, bone, and connective tissue. The endoderm forms into the epithelium of the gastrointestinal tract, the respiratory system, and many organs. Further differentiation of mesodermal cells begins the formation of the notochord. The notochord is important in starting or inducing the specialization of other cells, including those directly above them, called the neural plate. Epiblastic cells in the neural plate are induced to begin to mound up and fold into a groove. The neural groove continues to fold. Adjacent notochord mesodermal cells are induced to form somites. Somites are the eventual source of cells that form muscle tissue, skeletal, and other connective tissue. The neural groove completely folds inward to form the hollow neural tube. The neural tube arises and its cells eventually develop to form the brain, spinal cord, and other parts of the nervous system. Also during the 3rd week, mesodermal cells from the primitive streak rearrange to form blood vessels in the embryo, and a primitive heart tube is formed. Continued development of the chorion results in the formation of the chorionic villi, projecting into the endometrium to form an exchange area called the intervillous space. The maternal blood vessels and fetal blood vessels remain separate, so maternal and fetal blood do not normally mix. However, oxygen, antibodies, nutrients, and waste products are all exchanged via the intervillous spaces. During the 4th week, through a process called embryonic folding, the 3 flat layers of the trilaminar disc begin to fold around to form a 3-dimensional cylinder-shaped embryo. During this week, most organ systems continue or begin to develop via the process known as organogenesis. The nervous system and chorion continue to develop. The heart and the rest of the cardiovascular system continue to form. The digestive system begins to form. Portions of the entire digestive tract are now present. Ears, eyes, and limb buds begin to develop. The 2nd month of development is characterized by rapid development of the head and limbs, as well as continued organogenesis. During the 5th and 6th weeks, growth of the brain, and therefore head, is rapid. Hands and feet begin to form. During the 7th week, even more development of upper and lower limbs occurs. individual digits begin to form. By the end of the 8th week, the limbs are complete. Digits are separate. The tail disappears. The eyes are open, and the external genitalia are beginning to differentiate. Blood cells start to form in the liver. Bone ossification starts. The end of week 8 marks the end of embryonic development. While the embryo is only less than an inch in size, it now has some distinctly human characteristics. It is now referred to as a fetus. The fetus continues to grow during the 9th through 12th weeks. The head represents about half the length of the fetus. The eyes and ears are visible. The heart has a detectable heartbeat. The kidneys have developed sufficiently to form urine. External genitalia are detectable. The fetus exhibits weak but undetectable body movements. By the 12th week after fertilization, the placenta, the organ responsible for nutrient and waste exchange between the fetus and the mother, has begun to produce essential hormones. In the 4th month of fetal development, the face looks more human as the eyes and ears move to their final positions. Organ systems continue to rapidly develop. The gender is usually determined easily now. The fetus grows to a length of about 6 or 7 inches and weighs about 4 ounces. By the end of the 5th month of development, the fetus grows to weigh about 1 pound, and fetal movements become perceptible. Fine hair, called lanugo, forms over the fetus. The head becomes less disproportionate as limbs and trunk grow. By the end of the 6th month, the fetus starts making some surfactant in the fetal lungs, and it begins to practice breathing movements. The eyes are almost completely developed, and the fetus can respond now to noise outside the uterus. During the 7th month, the fetus increases in weight to about 2 to 3 pounds as it gains body fat. Testes begin to descend. Many infants born prematurely at this time do survive with intensive medical support. During the 8th month, the fetus continues to accumulate subcutaneous fat and reaches a weight of about 5 pounds. The fetus usually assumes an upside-down position at this point in preparation for birth. The fetus begins to practice sucking, and the mother may begin to notice the fetus has a pattern of sleep and wakefulness. In the 9th month, much of the fine lanugo hair is shed. The brain continues to develop. The fetus now weighs 7 to 8 pounds and is about 20 inches long. About a quart of amniotic fluid surrounds and protects the fetus. In the span of only 9 months, a fertilized oocyte develops from a single cell into a mature 8-pound fetus, ready to leave the womb and begin life on its own.